So now that you know the organization of your DNA, this idea of going from your genome to chromosomes to genes and the nucleotides that make them, you should know that in April of 2003, the Human Genome Project was completed. And just to take that very complex process and make it a little bit more clear, that's where geneticists, biologists across the globe mapped which genes are where. That's basically them figuring out which recipe was found in which section, which chromosome. Addresses, if you will. Where did this gene live? Where does that gene exist? Which chromosome? So what did we learn from this? Your genome is a little over 3 billion base pairs, at least for your haploid genome, which is found in your gametes like oocytes and sperm. And from this, scientists figured out that there are about 20 to 25,000 encoding genes, genes that lead to proteins. And that means there's a lot of other DNA material that doesn't seem to code for anything. And so scientists called it junk, and I wish they wouldn't have done that. They said it was a lot of false genes, pseudogenes, repeating elements of DNA. And actually, since then, we've learned a lot of the human genome isn't junk at all. Some very clever scientists said, well, if it's junk, if I delete it from the genome, the cell should still function, and organisms should still result. And what we found is, no, that's not true. So it's not junk. And that's a fascinating story that's beyond the scope of this class. But if you want to look up what RNAs are and RNAis are, that's a really fascinating story. Or you can just come to me and talk to me about it, and I'll fill you in on that. But just suffice it to say, the rest of the human genome is not junk after all. Well, what are the letters used in the genes, the recipe? What letters are used? Well, they are actually quite literally letters that abbreviate these nitrogenous rings that I'm showing you in the top left. These rings can be purines, such as adenine and guanine, which are double nitrogenous rings, or pyrimidines, which are single nitrogenous rings, and these are cytosine, thymine, and uracil. If I link these purines and pyrimidines together, I can make a chain of genetic material. And if I have two chains that are kind of coiled around each other, shown down here, I create DNA, which stands for deoxyribose nucleic acid. It's a macromolecule, which means I take monomers, purines and pyrimidines, link them together to form a strand. And the strand has to fold. Very similar, isn't it, don't you think, to what you just learned about proteins. So DNA also is a linkage of monomers and forming polymers, and these polymers have to interact with each other. So DNA is double-stranded. And these two strands that you can see here actually have opposite orientation. We say they're anti-parallel. And scientists, being scientists, they have quirky ways of saying this anti-parallel. They say there's a 5' end and a 3' end, and the other strand orientates with its 3' end next to the 5' end. It's just anti-parallel, sort of like a yin and a yang, sort of like Velcro, right? There's a prickly side of Velcro and there's a soft side. You can't get functional Velcro if you have two soft sides. They don't interact with each other. So there has to be opposite orientation. And there are rules to DNA synthesis. There's base pairing rules. A always goes to T and C always goes to G. And you should know that base pairing rule. And the overall helical structure of DNA was first conceptualized by Watson and Crick, shown here, but they actually did their experiments in the laboratory of Maurice Wilkins, and the lab technician that helped take X-ray pictures of DNA was Rosalind Franklin. So all four of these individuals should be credited for the DNA structure, or at least deciphering it and figuring it out. Sadly, Rosalind Franklin died 4 years prior to the group receiving the Nobel Prize in science for the discovery of this DNA structure. And the Nobel Prize cannot go to someone who's dead. But we definitely need to give credit to all 4 individuals for this, not just Watson and Crick. So DNA and RNA are polymers of monomers, just like protein is a polymer of monomers of amino acids. DNA, you heard me say, is deoxyribose nucleic acid, and RNA is ribose nucleic acid. Well, ribose is a 5-carbon sugar, and you can see a picture of a ribose sugar down here, and you can see deoxyribose up here in this DNA nucleotide. And the difference between them, if you look very closely, is deoxyribose. Deoxy means it's lost a hydroxyl group. Down here, you can see that OH hydroxyl group on the RNA. DNA, you heard me say, is double-stranded, 2 strands that coil around each other. RNA is single-stranded, only 1. And another difference between DNA and RNA is that instead of thymine, uracil is used. And this is a very important substitution. Uracil is used. Well, how do we go from DNA to RNA? And this can be very complicated in the lecture. It can be very simple. I'm going to choose the simple. What do you need to know for the test? DNA is precious. It is your official recipe. You don't want it leaving the nucleus. It needs to stay in the nucleus. 99% of your DNA is in the nucleus. 1% is found as mitochondrial DNA in mitochondria. But in both places, it's contained. You need containment. It's precious. Precious documents. Just like your passport, you keep it in a very special place. You don't want to lose it. So DNA is your precious, precious working code. RNA, shown here, is you making a replica, a transcript. Messenger RNA is your transcript of DNA. And it's single-stranded, and this process of transcription occurs in the nucleus. So RNA is another macromolecule. It consists of individual nucleotides shown here, linked together. It's single-stranded, not double-stranded, shown here in the helical formation of DNA. Shown here, the DNA unwinds enough where an enzyme called the RNA polymerase can link together nucleotides to make a copy. And again, instead of using deoxyribose, it uses ribose. And instead of using thymine, it uses uracil. So we've made a transcript. We made a copy of our DNA. The code has changed a little bit. It's single-stranded, uses uracil, and the sugar is a little different. So we're changing the code, but it still is a working copy. After that, the RNA, shown here, is going to leave the nucleus and go to the cytoplasm. Well, there has to be some modification of that RNA. There are some parts of that long strand of RNA that we don't really need. So those parts of the RNA that we don't need, shown here, are called introns. We're going to splice them out. And the parts that we need, we splice them together. So you don't need to worry about that process. I just want you to know that there is some processing, like an editing function. There's some specialized modifications that occur to the RNA. Because it is single-stranded, we need to know which side is the beginning side, which side is the end side. So there's these things called guanine caps on the 5' or beginning end, poly A tail on the 3' or the end part of the RNA. And then finally, the end product can leave the nucleus and go to the cytoplasm. Well, once the modified messenger RNA, our transcript, our copy of our precious document that we call DNA, once it goes to the cytoplasm, now what do we do with it? How do we turn it into protein. How does this code for a protein? This is a process of translation. Translation occurs in the cytoplasm. Translation means we're converting RNA, or using it, I should say, as a code to synthesize protein. We're not converting RNA to protein. Please do not say that. That's not what we're doing. What we're doing is we're converting a code of RNA into a new code, a new structure called protein. So this messenger RNA has to be processed using a ribosome, shown here, this hamburger bun looking thing. It has a large unit and a small subunit. And it has structures inside called the A site and P site. I like to think of them as the arrival site and the polypeptide site. And there's even actually another site called the E site where it exits. Anyway, so this ribosome kind of glides along the strand of the messenger RNA. And what comes into the arrival site? Well, we need another kind of RNA called a transfer RNA, shown here. And a transfer RNA is going to bring into the ribosome the amino acids. Well, how does it know which amino acid to bring? Well, that's all dictated by the code here shown in the RNA. Let's explore that more. So here's our ribosome, large subunit, small subunit, gliding along. And the ribosome is going to monitor, detect 3 nucleotides at a time. And that code is called a codon. And so 3 nucleotides at a time dictate which tRNA will come with which specific amino acid. And as you can see down here, as the ribosome reads 3 more nucleotides, a new transfer RNA with another amino acid will come into the arrival site, the A site. You can see here the growing polypeptide chain is now linked to the new amino acid. Once it's linked, that old transfer RNA is released by the ribosome. And this transfer RNA now moves into the P site to allow a new transfer RNA with a new amino acid to arrive in the A site. So again, translation or protein synthesis proper occurs in the cytoplasm, and it is basically taking the code of RNA and using it to generate protein. The messenger RNA dictates the order of the amino acids and which amino acids to use. This process of translation has been evaluated, and we now know the code of the 3 nucleotides that are used in a codon and which amino acid is used, as shown here. We call this the universal code, although it's not wholly universal. There are some exceptions, as there usually are to anything in this life, but it's pretty universal. We know that some nucleotides, when arranged in a codon, will tell the ribosome to fall off. We call these stop codons. We know that there are several different codons that can lead to the same amino acid. We call that redundancy. That's actually a very protective thing in biology. So there's redundancy. There's punctuation, basically. It tells the ribosome when to begin, when to stop, and even when to insert an amino acid that causes a protein to start to fold in a certain way. Very interesting. In summary, gene expression means we're going from DNA to RNA. That's transcription. It occurs in the nucleus. Then RNA to protein, shown here with the amino acids linking together. That's called translation, and that occurs in the cytoplasm. Why do we care about gene expression? Again, that's what tells a heart to look and behave like a heart and a brain to look and behave like a brain. and what makes you different from me. And our gene expression changes. This is a very fluid process. Simple touch, me touching your hand, can change your gene expression, can lead to hormones being released that change your gene expression. Smelling coffee, for example, even changes gene expression. Some people are more depressed than others. Why is that? Changes in gene expression. Everything you can think of in medicine, how a patient reacts to a medicine, and why one patient reacts to it and another one doesn't. That's all gene expression. It's all gene expression. It's all this idea of turning on and turning off genes. And yes, your cells have that ability. They can turn genes on, turn genes off, and that is very remarkable. Last slide, I just want you to know a little bit of terminology. Mutation versus DNA damage. Those are not synonyms, and a lot of people think they are, even some of the biologists that I have worked with. DNA damage means just that. There's been damage, and it can be corrected. And if it's corrected, then there's no harm. There's nothing residual. You can think of that as your floors are dirty, so you clean them. They're still your floors, right? There's been nothing changed to them. When damage becomes a mutation is a totally different thing. Damage that's not repaired can lead to a mutation. For example, uracil. Uracil is supposed to be found in RNA, but uracil can be found in DNA. Uracil that's found in DNA, it's not supposed to be there. So when it gets inserted in DNA, it's a nucleotide, and it happens quite a lot actually. That's damage. If it's not repaired, if it's not cleaned up, uracil can actually code for a different nucleotide if that part of DNA is transcribed into a messenger RNA. It'll change the code. Now that'll lead also to changes in the DNA strand, because again, you have two DNA strands that have to combine together, right? So uracil, if not corrected, if not cleaned up, can lead to a change in the code. Now, anytime you have a change in the code, that is now officially a mutation. And that change in the code has to be official. It has to be transferred on to the next generation of cells. If we're talking about mitotically active cells, cells that are dividing, it has to be transferable to the next generation. And that can even be literally in the next generation of the human. If a woman is pregnant and going to give birth, that's the next generation. She can transfer on that mutation to her child. What's this idea of totipotent? A totipotent cell is a cell that can create a brand new organism plus all the extra embryonic membranes, like the placenta and the amniotic sac, just to name a few. So what is a totipotent cell? Best example is a zygote, where sperm and oocyte unite and the nuclei fuse. That is a totipotent cell. It's going to create a brand new organism plus the membranes for that organism to bind to the mother's uterus. Pluripotent. If I'm talking about a pluripotent cell, that cell can only generate a brand new organism. The best example of that would be a blastomere from a blastocyst. Those of you who have already taken anatomy, you remember a blastocyst is a spherical ball of cells with a hollow center and an inner cell mass. If I were to take a single cell from the inner cell mass, that cell can generate a brand new organism, but it cannot create the extra embryonic membranes. Multipotent. A multipotent cell means it can generate many different kinds of cells but they have a common link. For example, the best example is a hemocytoblast. A hemocytoblast is shown down here. Its daughter cells start to progress and differentiate and become very unique, but they're all going to be cells that are involved with blood. So a hemocytoblast will give rise to platelets, gives rise to all your white blood cells, gives rise to your red blood cells, but they're all factors that are found in your blood. Unipotent cells. These are cells that replicate and divide for your entire lifetime, but they only can give rise to one kind of cell. One example would be this proerythroblast that I've shown you that comes from a hemocytoblast. Another example would be sperm, right? A man can make sperm for almost his entire life, almost, once he hits puberty. But those stem cells only give rise to sperm. Well, so what is a stem cell? These terms that I just taught you, totipotent, pluripotent, multipotent, unipotent, these are cells that can replicate and divide and give rise to other cells. A totipotent cell will replicate and divide and give rise to a brand new organism plus extra embryonic membranes. A pluripotent cell can replicate and divide, give rise to a new organism. Not every stem cell is as powerful as the next. A unipotent stem cell can only replicate and divide and give rise to one kind of cell, not a brand new organism. And this tells us that there are different stages of progression for a cell's life. There's a stage where it's determined versus differentiated. A determined cell is a lot like you. You're in my class because it's a prereq for some other class. You're determined. You woke up one day and you said, I'm gonna be most of you are going to be nurses or want to be nurses. So I'm going to use that as an example. You all said, I'm going to be a nurse. All right, you're determined. You're determined to be a nurse. But I also have people in this class who want to be physician assistants and dental hygienists and dietitians. And when I look out at all of you, I can't differentiate which of you want to be nurses and which of you want to be PAs. I have no idea. You all look the same to me. Well, scientists can collect cells from an organism. We do this all the time. Look at them under microscopes. And I can look at two cells and I can't tell the difference between them. But yet, on a molecular level, one might have awakened, if you will, one day and said, I'm going to be a heart. And the other one said, I'm going to be a brain or a neuron in a brain. They're both determined, but they have not finished the gene expression. They have not selected which genes to turn on, which genes to turn off. They look no different to me. When they finish their maturation, when they go through all of the gene expression they need to, they become specialized. And when a cell is specialized, we say it's differentiated. And when a cell is differentiated or specialized, I can look at a neuron under a microscope. I can look at a cardiac cell under a microscope, and I know instantly what they are. They look different. They are specialized. And the proteins they turned on allow them to have different shapes, allow them to have different functions. Just like you, all of you, you woke up today determined to become something. But to me, right now, you all look the same. Years from now, maybe 2, maybe 3, you might come back to visit me, and you might be in your specialized uniform, and I might be able to look at you then and say, ah, yes, I see the stethoscope around your neck. I see the white coat and the scrubs. Or I might say, ah, yes, I see your dental utensils to hand scale someone's teeth. I know what you are. I can see what you are. I don't have to ask you. That means you're differentiated. You're specialized. And that concludes our lecture on gene expression.